Hello, I'm Paul Joseph D. Mendoza, a cell and molecular biologist, and here are what are we usually doing inside the laboratory in our everyday life. In the laboratory, we study about the cellular molecules, organelles, cellular structures, and different processes that happens in cells and molecules. Our work focuses on many things, from studying different kinds of bacteria and viruses and developing cures and vaccines for the diseases and illnesses they are causing, or in development of therapeutic drugs, and even in the research related to stem cells, and a lot more related areas in the field of medical development. Our job also focuses on researching and experimenting about cell growth and its development, like cloning, gene repairing, editing, and expression, and other related to cell and molecular biology laboratory works. Now, here are some of the basic tools that are existing inside the cell and molecular biology lab. Beakers. These are used to retain liquid or solid samples or as a container for reactions. They are also used to collect filtrates from filtering process as well as liquid from titrations. Watch glasses. These circular glasses are used for holding small samples and for covering the beakers. Test tubes. These tubes are used in a lab to hold, mix, and apply heat to small amounts of chemicals and samples. Test tube holders. These holders are used to hold the test tube during experimentation. When touching the test tube is hazardous. Erlenmeyer flasks. These flasks are useful for holding liquid substance or for containing reactions. They are helpful for catching filtrates as well. Pipettes. Pipettes are used to dispense a small and accurate amount of liquid inside the lab. Bunsen burner. These are used for the source of heat inside the lab. Now, here are some of the common equipments that we're working with inside the laboratory. Balances. These are used for determining the mass of the reagents and the objects inside the laboratory. Spectrophotometers. These are used to measure a liquid sample's absorbance or transmittance. Fume hoods. These are used for ventilation of hazardous or harmful gases. As a cell and molecular biologist, effective analysis of various data is needed constantly in order for us to produce an accurate results and researches. Now, here are the examples of techniques that is being used inside the laboratory. Cell or tissue culture. Similar to how bacteria and other simple creatures can be cultivated in a lab environment, other than their natural habitat, cells and tissue from more complex organisms can also be cultured inside the lab. Bright field microscopy, the simplest optical microscope illuminating method. White light is utilized to illuminate the sample, and contrast is created in the sample by the sample's dense portions attenuating the transmitted light. RNA interference. By attaching the target sequence and blocking the translation, the interfering RNA interferes with the target sequence. Therefore, a specific gene can be silenced by using an interfering RNA that has been carefully chosen. Overall, the life as a cell and molecular biologist is not an easy life, but the ability to help humanity in many ways, and also using my time here on earth for the extension of knowledge of humanity is a fulfilling and a life well spent. Thank you and goodbye.